Hello everyone. Today we are going to discuss about the phylum Porifera. So that is the first chapter in the second unit of the paper Animal Diversity 1, that is non-chordates. So the second unit, it consists of phylum Porifera and the phylum Cylindriata. So in this chapter, that is phylum Porifera, we'll be studying about the general characteristics of the phylum, classification up to their classes level, and histology of spicules and canal system in sponges and systemic position of Porifera. So in this session, mainly we'll be discussing about the general characteristics and classification up to their classes. So the poriferans are nothing but they are animals which are having pores on them. So as they can be defined as, they are asymmetrical or radially symmetrical multicellular organism with cellular grade of organization but without well defined tissue or organs. So they are exclusively aquatic, maybe marine or freshwater. So the name sponges was given by the Elias, and Robert Grant, he was the person who termed the Porifera, and Elias was the person who given or he proved that the sponges are animals, and the study of sponges is called as parazoology. So let's begin with the general characteristics. So we know that Porifera, as you can see in the picture, the sponges which are mainly present in aquatic habitat, they all are aquatic. Maximum of them are marine and some of them are the freshwater. Example is the family Spongillariidae. So these are sessile and sedentary. So as you can see in the picture, in the deep oceans, they are attached to the substratum, maybe on a plant or maybe on a stone. So they are sessile and sedentary, and they usually grow like plant. They branch up, and the body shape is somewhat vase-like, cylindrical, and if you cut the sponges into half, it may be symmetrical or radially symmetrical, or asymmetrical, radially symmetrical or asymmetrical. So the body surface is perforated. So as I said, it consists of numerous pores. So that numerous pores is called as ostia, through which what happens, the water enters inside and moves outside. So that water exits through a larger opening on the top of that is called as oscula. So ostia is nothing but the small numerous pores. I will show in the picture. It is a multicellular organism and cellular level of body organization. So there is not much development, so that cells are organized together to form a tissue. So here there are no tissues, only cellular grade of organization you can find. So they consist of outer ectoderm, inner endoderm, but intermediate layer is called as mesenchyma. So if it is having two layers, so in the previous unit of the introduction I have said, if they are having two layers, then it is called as diploblastic animals. So these poriferans are diploblastic in nature. So this is the structure. You can see the water, it is moving out through the osculum and it is entering into, that is called as ostia. So it is a cross section of sponge, how the water enters inside and how the water is thrown outside. So you can see the outer ectoderm and inner endoderm, and middle layer, that is mesochyle or mesenchymal layer. And also you can see the spicules. So what are these spicules now? So spicules we will be studying in detail. They are the important structures which provide the structure to the sponges. So because they don't have any type of structure for their giving organization or giving rigid body structure. So these spicules helps in giving rigid body structure. So next, so as you can see the same picture, how the water is entering into the ostia and how the water is thrown throughout by osculum. Though this happens inside the sponges. So even this is the same thing, how the differentiation of endoderm, ectoderm and mesenchymal layer is present. So just make a note on these structures, they may be asked for your examination. So next, the interior space of the body is either hollow, as you saw in the picture, the internal structure will be hollow in nature. So there what happens, there the water will come inside and through osculum it is moving outside. But these spores, what they are lined up by term numerous canals called as kinetocytes or conacocytes. So the interior space of the sponges is called as spongocoel. Very important terminology. Spongocoel is nothing but the interior space of the sponges where water enters inside and exits out. So and as I have said, they consist of the spicules or the spongin fibers. So characteristic skeleton, they are made up of mainly calcium. So then only the rigid structure of the sponges is seen. So these may have spongin fibers or the calcareous spicules or siliceous spicules. So calcareous, it is made up of calcium, siliceous, it is made up of silicon. So mouth is absent because there is not much organized tissue or organ formation. So digestion is intracellular because it happens within the cell, and excretory and respiratory organs are absent, but it takes place through a specific wall structure. So contractile vacuoles are present in some of the fresh forms. The nervous and sensory cells are probably not differentiated because the cells are not much evolved. For that reason, the nervous and sensory cells are not differentiated. And nervous system, as I have said, so here you can find bipolar and multipolar cells which take the signal or which receives the signal from the external environment. And sponges are monoecious. So what do you mean by monoecious? They are made up of single type of cells. So they are called as monoecious. And reproduction, it occurs both sexually and asexually method. So again, in asexual, we are having the bud formation and gemmules formation. The sponges possess the higher power of regeneration. So if you cut the structure of the sponges at the tip of the sponge, so it will not die. It has a tendency to regenerate, like maybe a tapeworm or a planaria you have studied in your lower classes. Even these sponges, they are having their regeneration power. Next, sexual reproduction, it occurs by production of ova and sperms, and all sponges are hermaphrodite. It means both the sexes, may be male and female, they are present within the same body. And fertilization is internal, but cross fertilization can occur, and cleavage will be holoblastic. So the development here, what happens? It takes place by the free swimming ciliated larvae. So these larvae, very important, what they are called as, they can ask you for 1 mark. So amphiblastula. So what do you mean by amphiblastula? It is a development in indirect through the free swimming ciliated larva of poriferans. They are called as amphiblastula larva. And you can find the organization of sponges in different types. So in the picture you can see asconoid, syconoid, leuconoid. So these are the 3 different types of the sponges formation on the basis of or the arrangement of cells. So example, do remember sycon, Eupoleptala, then Sponginia. So at least remember 3 to 4 examples. So next we'll move on to the classification of the sponges. Here we are having only 3 classes. So they are Calcarea, Desmospongia, Hexanectillida. So under that, we'll be studying in detail. So it can be asked for 5 marks about their classification with their example. So this classification of sponges is mainly based on the type of skeleton and the cells which are arranged inside the sponges. So which was given by Hyman in 1940, and later it was modified by Bruton in 1967. So later they classified as first class, that is Calcarea. So Calcarea means what now? So these sponges, they consist of the lime. So what we call that is lime, they are having that content inside their cells, or they are made up of that type of lime. So they are classified the name with the Calcarea. So the characteristics of this class Calcarea are, they are having the skeleton of separate calcareous spicules. So I have mentioned about these spicules. Spicules are nothing but they are the rigid forms which helps in the development of the skeleton, or which gives the rigid structure to the sponges. So these spicules are made up of calcareous, and these spicules are of monoaxon, tetraaxon or tetraaxon. So we'll be studying about these spicules in detail in the next class, but remember the name. Monoaxon means the spicules which are having only one end, or one pointed end, and one will be blunted. Tetraaxon means all the 4 sides of the spicules will be pointed, or 4 sides, that is tetraaxon. So we'll be seeing in detail, and they are solitary or colonial. So they may be live in large colonies, or they are single in nature, and their body shape is somewhat vase shaped and cylindrical. They may show all 3 types of variation, that is canal formation, that is asconoid, syconoid and leuconoid structures. And usually these calcarea, they are dully colored sponges, and they varies around 15 centimeters in size, and these occur very shallow in deep water, very shallow on water in oceans. Examples are Sycon. So this is about the class Calcarea. So next, moving to second class, that is Hexactinellida. So here Hexactinellida, usually the other name is Hyalospongiae. So hyalose means it is glassy. So you might have seen in the lab the Eupaleptala. So that is the example. So they are glassy sponges, and skeleton, it is made up of siliceous. So silica is somewhat silicon material are somewhat transparent, and hence they occur glassy nature or white color in nature. So these glassy sponges or Hexactinellida, they are made up of siliceous spicules which are having triaxon. So tri means 3 with 6 rays, means triaxon, 3 pointed spicules are there, but they are arranged side by side. So 3 plus 3, 6. So it is called as Hexactinellida. So it is having 6 rays. And there is no epidermal epithelium. And conacocytes, as I said, they are nothing but the cells which are present in inner layer of the sponges, as you have seen in cross section of the sponges. And these are cylindrical or funnel shaped and found in deep tropical seas. So previously Calcarea was seen in shallow, but here these Eupaleptala or the glassy sponges you can find in deep oceans. So they can grow up to 1 meter height also. So this is about the second class, that is Hexactinellida. Next, class 3, Demospongiae. So in name itself, Demospongiae means it is dermose, means the frame, the sponge. Again, it consists of large number of sponge species. So these are having larger in number, and skeletal, they are made up of spongin fibers. So here, if they are made up of spongin fiber, means more number of the ostia or the pores will be there. So these spongin fibers with siliceous spicules, they may be having no skeleton now, because they are having more pores, so that gives the rigid structure now. And spicules are never 6-rayed, because 6-rayed will be always only hexactinida, but here they are monoaxon or tetraaxon, and they are differentiated into larger megasclerates and smaller microsclerates. And body shape is irregular, and canal system only you can find is leucon type. Generally they are marine, usually few are freshwater. Example is spongella. So as you can see in the picture, they are widely branched, and height will be smaller. The much larger will be hexactinida, and you can find them in deep sea. But these two, Demospongia and Calcarea, you can find in shallow water. But more number of spores are there, then it is Demospongiae, because they are having spongin fiber, and less number of sponges, it will be the Calcarea. And usually hexactinida you cannot find on shallow water. You have to go deep into ocean, then you can find very large, up to 1 meter in height also. So in this session we have studied the general characteristics of Porifera and their classification. So that's all about this session. Thank you.